Our next talk is going to be by Professor Dr. Guzman, Raphael Guzman, from Switzerland. His topic is going to be neuroregeneration in stroke, 20 years in the making. Dr. Guzman is the chair of neurosurgery at University Hospital in Basel, Switzerland, where he focused on cerebrovascular and pediatric neurosurgery. He was a neurosurgery resident in Switzerland, in Bern, Switzerland, then completed 2-year fellowships at Stanford University in open vascular neurosurgery under the guidance of Professor Steinbeck, and also pediatric neurosurgery under the guidance of Professor Edwards. He was appointed assistant professor of neurosurgery at Stanford before he moved back to Switzerland in 2012. Dr. Guzman's research started with the thesis in neuroregeneration for Parkinson's disease. Thereafter, he focused on stem cell mediated regeneration and neuroproliferation in stroke, which is still the topic of his current research in the Department of Biosciences in Basel. Please join me in welcome Professor Guzman. Thank you.
Yes, thanks a lot for the kind introduction, and thanks, Daniel, for the invitation. It's a great pleasure to be here. Always nice to visit New York, of course. Very nice to see old friends. We will change gear a little bit, I think, from health politics more to research. But I hope that I can guide you through what has been going on in stroke and regeneration over the last 20 years. It's going to be 20 plus last year. And I hope that by the end of my talk, I can give you some concepts. What are the current thought processes involved in this very difficult topic? And I don't think with this crowd that I really have to point out the global burden of stroke. 101 million people living with stroke aftermath, and 1 in 4 at the age of 25 will suffer a stroke over a lifetime. So we're very clear that stroke poses a big problem for society, and we've been talking about it. Most of all, actually, in the last talk, that was fascinating. The awareness is in the society is so important to improve stroke care. And if we look at the demographic evolution, and here you have actually a forecast for New York in 2050, the stroke incidence with the aging population will actually significantly increase, just augmenting the problem that we will face in the future. And therefore, like here, we've just heard it, in Switzerland as well, we have started what's called the Swiss Brain Health Plan 2023 to 2033. It's a big consortium of clinical neuroscientists that came together. And the plan is really to raise awareness about brain health in general, stroke included, but also traumatic brain injury and dementia, but also very important for all psychiatric disorders. So mental health is a huge problem that is included in this brain health plan. Interprofessional training, extremely important. Research, and that's where I will focus today, but research on brain health determinants, on improved care, both in prevention, but also in treatment. Brain health promotion and empowering patients and caregivers to really adhere to the plan and hopefully make it safer to live into the next decade. So this is a big initiative that is supported by politics, just like was mentioned in the last talks. Extremely important to have the politicians on board to open doors and to allow to actually have such a healthcare plan communicated to the society. So that's how Switzerland is organized. Switzerland is small. You will see here 220K by 348K. That's about from here to Boston. That's Switzerland, 8 million people. But this is an advantage in the sense that we can have a very tight network of stroke centers and stroke units. The area that is kind of blank is because it's mountains. So it's not because there's no stroke units or centers, it's just mountains. But you see where the population of Switzerland lives. what's called the Midland and the northern part and the western and the eastern part of Switzerland. And we still have about 15,000 strokes per year. That's probably what Brooklyn has. But at the national level, of course, it's very important. And with this tight plan, actually, we managed to have every patient be in the hospital in a time frame that can get IV thrombolysis and then intraarterial treatment. So it's the advantage of a small country with a very tight network. Now, despite very good treatment, we know that we will have 2/3 of the patients in Switzerland, but everywhere else too, leave the hospital with a disability. More than 50% of the patients who had a stroke will be left dependent on others for everyday activities. And this is despite best treatments that we have these days. And that's why there is still a huge need for additional therapies, because we have, of course, the IV treatment and the intraarterial thrombectomy, thrombolysis treatment. We have excellent rehabilitation, but we need other treatments to cure or at least to improve quality of life of those patients. As you might know, over the last also 30 years, there has been tremendous research going on in trying to develop drugs. And we really have this pharmacological graveyard of billions of research dollars spent by universities, by big pharma, that have actually, unfortunately, not led to the successes that we were hoping for. And this is just a little list of some of the drugs that have been tested and have never made it to clinical application, because it was found to be extremely efficacious in the animal studies, but actually never found to be efficacious in patients. And if we just look very briefly at some of the pathophysiological pathways involved in stroke, where we have an energy failure, and then excitotoxicity with glutamate release, a problem with sodium-potassium channels that stop working, which will lead to internal depolarization in the cells, mitochondrial damage. We have radical oxygen species that will lead to a very strong inflammatory response that starts actually very early, 15 minutes after onset of stroke. We will start having inflammatory response with an upregulation of interleukin-1 alpha to start with, and then beta, and then all the other factors, and then culminating in a full-blown inflammatory response after 48 to 72 hours. Now, for all these, drugs have been tested. And as I mentioned, preclinical, extremely successful. So, for instance, the NMDAR blockers that have been very successful in rodent studies and large animal studies, but in clinical trials actually have failed to demonstrate the benefit. All the sodium and potassium channel blockers, blocking here, then anti-apoptosis measures, anti-inflammatory drugs, and so forth, radical oxygen scavengers. All of those had a very good molecular pathway as a target, but never made it to clinical success. And so we're still left with no treatment after the thrombolysis that we have these days. And so one of the hypotheses or one of the goals that has been formulated is that when a stroke happens, after having the standard care that we have today, we should still try to develop neuroprotective agents. The neuroprotection would be very strong if we can start it right after stroke or within 72 hours. So the first part, or the acute part after stroke, is called neuroprotection. When we go to the subacute phase, we discuss intravascular cell transplantation, and I will talk about it. So this is partly neuroprotection, but partly also neuroregeneration. Hypothermia, for instance, is one of the strong neuroprotectants that has been tried extensively, that actually works in the neonates with hypoxic ischemic encephalopathy, but in the adults also has shown limited data and is not being applicated. But hypothermia is actually one of the extremely strong neuroprotectants. As we go to a more chronic phase, we talk again about intracerebral transplantation of stem cells, with a goal to have a neuroregeneration more than a neuroprotection. And here I will talk about this phase. So we go from a neuroprotective strategy to a neurorestorative strategy experimentally. So cellular therapies, and I will jump right into stem cell therapy as a field of research. Is it hype or hope? Is it a reality or not? And I think the opinion really goes apart. For a lot of it, actually, I think it's a hype. A lot of patients are coming even to my clinic asking for stem cell therapy, either of children with a cerebral palsy or adults that have suffered a stroke. So all that we read in the news and so forth still is kind of a hype. But of course, there's a lot of hope, and there has been a lot of hope over the last 20 years that stem cell therapy would come to fruition to actually restore brain function. So a small historical vignette going back more than 100 years. We had Sir Wilhelm His, who actually, I found out, came from Basel. So he was born in Basel, the city I'm coming from, the city Daniel and I have worked together, 1831 to 1904. He became a professor with 26 years old. Would never happen these days. He was a professor of anatomy and actually has written that book that was then later published after his death, Die Entwicklung des menschlichen Gehirns, so the development of the human brain. And very early on, he had done very extensive studies on the periventricular area, which is one of the neurogenic zones, one of the zones where actually neural stem cells reside and actually are there to form the cortex development. And if we go forward, we have the work of Wilhelm His that actually already shows the radial glia, a concept that is still true today. Radial glia with their extension. He even showed cells that are mitotic and then post-mitotic cells migrating from the periventricular area to the cortical surface. So very actually precise depiction of what's happening in brain development with stem cells, not knowing that it's stem cells. They were called spongioblasts. Retzius, using the Golgi method, and then famously Ramon de Cajal actually made the known drawings of the periventricular or subventricular zone where the radial glia and the endogenous neurogenesis is happening, both for the brain and also the spinal cord. So there's a long history. It's just that the stem cells were not actually known, but they were drawn, they were observed, and it was known that brain development must stem from a cell that is able to divide and then populate the cortex after birth. So this is a little bit of the timeline that we will go through in my talk. As we said, we started in the 1900s just by drawing, but not recognizing what it is. And then we went to the first preclinical and first clinical transplantation studies for Parkinson's disease. And that's when I started actually doing research in 1994, 1995 on trying to regenerate the dopaminergic neurons that degenerate in Parkinson's disease. And as you know, the degeneration of the dopaminergic neurons in the ventral forebrain, in the striatum, and in the mesencephalon were an excellent target for transplantation, because the thought was that if we can replace dopaminergic neurons producing dopamine in the striatum, we could actually restore Parkinson's patients in their function. And we started, you see, 97, 98, transplanting ventral mesencephalic dopaminergic neurons taken from fetuses, either rodents or then also humans, and culturing them in what we would call today organotypic cultures. Back then, it was just a bunch of cells. We had no notion that they were probably partially stem cells. To us, they were differentiated dopaminergic neurons. We were able to measure the dopamine release using HPLC and transplanted them in rats. And what we saw in the rat model, it was this MTPT or 6-hydroxydopamine models. We saw that animal function, which is a rotational behavior if they have an injury, could be restored after cell transplantation. And that's what it looked like in the brain, an integration of those cells and restoration of function. And we went on actually to develop those cells for cell transplantation in patients. So we had a GMP facility. They were called free-floating roller tube cultures, partially probably stem cells, but it was not known back then. And that's what they looked like, little balls that could be stereotactically injected into the patient's brain. And so excitingly, 1996, we were able to transplant the first patient at the University Hospital in Bern. That's the capital city of Switzerland, in this one Parkinson patient. And we were part of a big network where first patients were transplanted in Lund in Sweden and in the UK at Cambridge. And part of this network, a series of patients were transplanted with this technology. At the same time, there was a transplantation network in the US led by back then Ron Backay. And this was a big story. The unfortunate thing is that the cells actually did not survive. So some patients had a benefit for a short time, just like you have in deep brain stimulation, doing the injury in the putamen or in the subthalamic nucleus, but the cells did not survive. We know this now from longitudinal studies, and the patient symptoms came back. So the success that was preclinical proven actually never worked in the patient and still hasn't worked. But it was a big deal. First transplantation, 1996. We celebrated, of course. What else would you expect? 1996, that was the head of the lab that was developing the free-floating roller tube cultures, and that part of the lab team. I think we have aged reasonably well. We're still here and still working on it. So that's part of this 20-year journey. And I show you another picture here, also having a lot of beers after successful transplantation of the first patient. And back here you see a person called Lorenz Studer. Lorenz Studer ended up doing a postdoc in the US and actually became famous. He's here in New York. He's the founder and director of the Brainstem Neural Stem Cell Institute at Memorial Sloan Kettering here in New York. So he has made a nice career, and he's also partially or heavily involved in this New York Stem Cell Foundation Research Institute. So he was a postdoc or a PhD student in the lab where I did my PhD, and then became a big scientist name here in New York, closing the circles. So moving forward, actually 1992 to 95 was when the first description of truly neural stem cells actually came out. And there was the seminal work of several scientists, but most famously here Reynolds and Weiss, published in Science 1992, and then the group around Fred Gage at the Salk Institute in San Diego, really doing the first descriptions of neural stem cells and how these neural stem cells can actually be extracted, cultured, and grown in a dish and retransplanted. And this also was a seminal paper published in PNAS 1995, Survival and Differentiation of Adult Neuroprogenitor Cells Transplanted to the Adult Brain in Rodents. So this was really when the hype of stem cell began. And we can see, if we look at the number of publications on PubMed, you know, a long time not much happened because neural stem cells were actually not known to be there and nobody was able to culture them. But after 1996, there was a true increase, a significant increase in the number of publications related to neural stem cells and neural transplantation. The hype has a bit gone back, as you can see at the other end of that curve. So that was the foundation of starting to think about neuroregenerative medicine, both for the brain, but other organs as well. And this is the famous story, Greek mythology of Prometheus, who was actually punished by Zeus because he was stealing the fire. And the punishment that he was actually chained, sometimes it's chained on a stone, chained on a tree. And the eagle would come every day and pick his liver. But as you know, the liver actually has the capacity to regenerate. This we know for a fact. The liver was growing back every day. So this was the internal punishment of Prometheus back in the Greek mythology. And that's how regenerative medicine kind of was started, which is the process of replacing or regenerating human cells, tissues, or organs to restore or establish normal function. So that was kind of the start. Now we are about in the time frame of the last 20 years. And so the stem cells, without going in too much details on basics on stem cells, but they have the capacity to self-renew and to recreate functional tissues of different lineages. And we differentiate the totipotent embryonic stem cell derived stem cells going to multipotent adult stem cells. So what happened politically at that time, and now this is exactly 20 years ago, the legislation in the US under George W. Bush actually decided that there was ethical and moral problems with using stem cells, especially embryonic stem cells. And Washington decided that they will support financially only 60 cell lines that had been established before, but there would not be the possibility to actually make new cells and do really research on neural stem cells and other stem cells. And as a reaction in California, there was the creation of the California Institute for Regenerative Medicine in 2004, supported by this Proposition 71 that was adopted by the Californian population. And there was suddenly $3 billion being there for research with stem cells. And this was a very big deal. And by chance, that's just when I arrived at Stanford into that hype of stem cell research. And the fear was really that if the country does not support with federal money stem cell researchers actually will leave the US and go to Europe or Asia where they're allowed to do research. So it was counteracting this with a lot of money. And you see what was written in here without going in all the details, but millions of children and adults suffer from devastating diseases or injuries that are currently incurable. And then a couple. Recently, medical science has discovered a new way to attack chronic disease and injuries. The cure and treatment of this disease can potentially be accomplished through the use of new regenerative medical therapies, including a special type of human cells called stem cells. So that was the view in 2004, very interesting. And this really led to a hype. The year after, actually, the same happened in New York. So there was the creation of the New York Stem Cell Foundation, with equally a lot of money that really hyped the research around stem cells. But I think this was a critical event because without that, probably some of that research would have never happened, at least in the US. So these are the cells that are currently being used both preclinically, but also in clinical use. So we have the hematopoietic lineage stem cells, most used the bone marrow-derived mononuclear cells, or BMNC. These are sorted or unsorted mononuclear cells. They express this marker CD34 on the surface, and they can be sorted and then cultured in vitro. Another cell type that is used a lot is the umbilical cord blood-derived stem cells, either from the blood or from the matrix of the umbilical cord. These cells that are also mesenchymal stromal cells, so you see they will come in both groups here, can be cultured, multiplied in vitro, and then transplanted in rodent models, but also transplanted to patients. And a lot of the studies on cerebral palsy in neonatal hypoxic ischemic encephalopathy, these umbilical cord-derived blood stem cells are being used in clinical trials. Here we have the whole group of mesenchymal stromal lineage cells, again, the bone marrow umbilical cord, and I think very interesting adipose tissue. Many of us have a little bit too much of that, and this can actually be transformed into stem cells as an autologous transplant source. So very interesting, and there has actually been a couple clinical trials now using adipose stem cells. And then the committed neural lineage stem cells, so these are the actual neural stem cells derived from fetal brains that can be transplanted into the brain. So the two big concepts that we're looking at over the last 20 years, but still hold true, is on one side, cell replacement, and on the other side, trophic support. What does this mean? With cell replacement, we actually try to replace cells that have been damaged or injured in the brain. These cells are supposed to integrate and take over function of brain cells that have died after stroke. Trophic support is a type of cell that does not need to integrate into the brain, but will actually just support the brain by secreting certain factors. And over the next couple of slides, I will show you a little bit the differences in these two hypotheses, how the brain can be repaired. So when stem cell transplantation started in the first part of the 2000s, the whole concept was actually cell replacement. So it really started by, we need to transplant neural stem cells in the brain. They will integrate and they will restore function. And you see here transplantation of human neural stem cells. These were human fetal-derived stem cells in the olfactory bulb. So they were transplanted intraventricularly. They migrated along the rostral migratory stream all the way to the olfactory bulb. And you see here a beautiful integration in the olfactory bulb, actually producing neurons, interneurons, and a neural network in this rodent brain. So they were truly actually integrating in the brain. You see here in another model, this is called the Shiverer model, where the cells were transplanted in a mouse model that lacks oligodendrocytes, so lacks white matter or myelin. And actually these transplanted cells, you see here the cerebellum, actually could be remyelinated by transplantation of those cells. Another capacity that was demonstrated is that the cells not only integrate, but actually can transport action potentials. So you see here integration of human neural stem cells in this animal model of white matter disease. And you can actually stimulate with microelectrodes and see the propagation, as you see here, the propagation of action potentials from one electrode to the other, which does not happen in the unmyelinated brain, but happens if these brains were remyelinated by transplanting human neural stem cells. So you had a true integration of cells. A lot of work was done in stroke, where here a model of middle cerebral artery occlusion leads to a stroke in the cortex of a rat, and cells were transplanted adjacent to it. Here a transplant in non-stroked animals. And what was observed is that these cells actually can migrate from your transplant towards the stroke area and can integrate into the network and start differentiating into neural cells, into oligodendrocytes or myelinating cells and astrocytes. So the cell, excuse me. So the cells were shown to actually truly integrate in the brain and lead to functional recovery. So in yellow here, you see a neurological exam that tests the forepaw movement in a stroked animal that has a hemiparesis. And you see here that 8 weeks after transplantation, you have a significant improvement in the contralateral paw movement. A lot of neurological tests were done and shown that the animals do recover. So this was truly cell replacement. Another interesting fact is that transplanted cells could actually lead to enhanced brain plasticity. Here a study of a colleague of mine where he actually demonstrated that you had integration of cells and a support of host cells to reform dendritic tree and axons. So it was supporting, but it was also developing in neurons that would actually lead to connections in the brain. And he measured regeneration of the corticospinal tract. Here. He measured the recovery of the corticospinal tract after transplantation of cells at the level of the cortex, at the level of the striatum, in the brainstem and the spinal cord. So he was able to actually trace how axonal outgrowth from the brain to the spinal cord was enhanced in animals transplanted with neural stem cells. So enhancement of brain plasticity, but integration also of those cells, making new connections all the way down to the spinal cord. Another very important mechanism of action was the support of angiogenesis. So neural stem cells that were transplanted into the cortex after a stroke were shown to actually lead to a significant increase in neovascularization and also in stabilization of the blood-brain barrier. So less leakage of the blood-brain barrier and more new blood vessels growing in on the side of the stroked cortex. And this new angiogenesis actually was then correlated also to functional recovery. And an interesting trick that was done in that study was actually to give Avastin. Avastin that we all know from clinical use was actually selectively killing the human cells that were transplanted. So not killing the cells, excuse me, blocking VEGF receptor. And when, upon blocking of the VEGF receptor with Avastin, the effect on angiogenesis was lost. So it means that those cells that were responding to Avastin, if they could not lead to angiogenesis, they would actually also not lead to a functional recovery. So a very elegant study published in 2011. So taken together, the cells did integrate in the brain, but also had what we call paracrine effects, namely, and I did not show that now, but doing immune modulation, so reducing inflammation, then actually increasing the blood-brain barrier integrity, leading to angiogenesis, so revascularization in the stroke area, and actually lead to a reduced stroke size or reduction of cortical atrophy. So if we conclude on this first part, which is cell integration, it was demonstrated that the cells can integrate and migrate, very importantly, but the mechanism of action was probably not only cellular integration and differentiation, but enhancement of neuroplasticity, angiogenesis, and immune modulation. So it was a mix of cell integration and trophic support. What was shown in lots of studies is that actually not a lot of those cells survive. Okay, so you transplant the million cells, but only, depending on the study, 0.1 to 30% of those cells survived. Despite that, it was observed that you have a functional recovery. So despite the limited integration of the cells, you have an effect. So there must be other mechanisms of actions. And one study actually out of UCSF, who did a transplant study in another disease than stroke, was able to do brain sections and demonstrated that you have very little cells surviving in the human brain, but actually seeing some functional effect. And that's where the new concept was created, which was that I wrote in this editorial, cellular stroke therapy from cell replacement to trophic support. So really the whole field has shifted from 2010 onwards, has shifted from replacing the cells to rather using the cells to have a trophic support, supporting the brain in its recovery, both neuroprotection and also neurorestoration, but not through cell integration. So one of the clinical challenges in cell transplantation, as you all know, is that a stroke is not often a small, tiny stroke where we can imagine that you can transplant cells just in the vicinity of the stroke. What happens if you have a large MCA stroke? We need to find other ways to deliver the cells than doing very small millimeter-sized deposits in the brain. So the idea came that we should start doing intravascular delivery. And of course, for all of you who do intravascular therapy for stroke, this makes sense. Why should we transplant the cells into a very tiny area in the brain if we can actually deliver the cells through intravascular injection to larger areas of the brain? And during my time at Stanford, we did a lot of studies on intravascular, both intravenous and intraarterial cell delivery, and found that the cells can actually home to the area of stroke, not randomly, but the stem cells actually carry receptors, just like inflammatory cells, carry receptors for chemoattraction and chemoadhesion. So there is an active chemoattraction of the cells to the stroke area through receptors that are well known. So the CCR2, CD49D, CXCR4 that respond to chemokines such as CCL2, that respond to cell adhesion molecules, and that respond to chemokine CXCL12. So there was an active recruitment. And we found that if we knock out CCR2 on the stem cell, or if we block CD49 or knock out CXCR4, the cells would not home to the stroke area. So it was an active transport of cells from the bloodstream to the stroke area. So this was used actually to do a whole bunch of different studies. One was biodistribution. So we were labeling the cells with a fluorescent tag, able to do bioluminescence imaging. And you see after a tail vein injection that cells that do express those receptors preferentially actually home to the brain and actually start residing in the brain. But if you block those receptors, the cells will actually not home to the brain. We're able to show with MRI, labeling those cells with iron oxide nanoparticles, that the cells would actually home to the brain. And at the same time, colleagues at Hopkins were able to do live MRI in a rodent model after injection of these stem cells in the carotid artery. So cut down on the carotid artery, like we heard this morning, injection of the stem cells directly into the carotid artery in the ICA, and the cells would home. We did a lot of studies on cerebral blood flow and ischemic events in these intraarterial stem cells. We're able to show that it did not lead to ischemia or blockage of blood vessels. Going fast forward, we demonstrated also functional recovery after intraarterial injection of those stem cells, significantly better than if the animals got saline injection, both for motor tasks, but also for cognitive tasks as this novel object recognition. So this was very interesting, and we started looking at what are the mechanisms of action. Why do these animals actually recover? And one of the effects that we saw is that we had a significant restoration of white matter tracts. And we did some extensive studies on staining white matter tracts here at 10 days and here at 30 days after stroke and cell injection. And we quantified these changes using histology, but also using diffusion tensor imaging. For the neuroradiologist here in the audience, you can see actually much denser white matter tracts in the cell-treated animals as compared to the control animals. We measured the FA values, and there was a significant change in the FA values in animals that received cells versus not. And several other groups also did the same. So there's a pack of data that can show that. What it actually did, one of the effects, how did it actually remyelinate, is that the cells actually were enhancing endogenous neurogenesis. So this just as one of the mechanisms of action. There was actually an increase in newborn neural stem cells that would differentiate into oligodendrocytes. So also here, without going in too much detail, we had actually an enhancement of endogenous repair mechanisms in the brain, one of the effects of the cells. So the cells did not integrate, but helped the brain recover. And to make it more complicated, we did some gene expression studies on the brain tissue and found differential expression again 3 days and 10 days after treatment, that if you do transplantation of cells, you have a significant change in gene expression related to cell proliferation, cell signaling, inflammation. So these were all paracrine effects. The cells did not integrate, but actually changed the environment of the rodent brain. And so that concept actually was picked up by many groups showing the same thing, that you have an enhancement of endogenous repair mechanisms after ischemia, but also in the periphery. Here a study that looked at traumatic brain injury that was able to measure in plasma after injecting cells intravenously that you had a change in anti-inflammatory cytokines, namely interleukin 10 and interleukin 4, in cell transplanted animals as compared to saline treatment. So very interesting, you have a peripheral immune modulation and you have a central nervous system immune modulation as a very strong effect of action. So the hypothesis we started building was that if we do inject stem cells, we have on one hand a direct effect on the microenvironment in the brain, partially remyelination, endogenous neurogenesis, but also we had a part which was immune modulatory, changing the function of microglia. Microglia as the brain resident immune cell that is extremely important for the homeostasis, and that can be changed when the stem cells are transplanted. So when you look at a stroked rat brain, this animal has a stroke here on the right side, and in red you have a staining of the microglia. You see that microglia are abundant in the subventricular zone, in the striatum ipsilateral to the stroke, in the dorsal end, which has both neurogenic zones, abundance of microglia. So it cannot be just inflammation. Maybe the microglia has another function than just doing neuroinflammation. Maybe the microglia has a positive effect. And it is known since 2017 that microglia can actually adopt a pro-neurogenic phenotype. We have this what's called M1 and M2 phenotype of microglia. So we were interested, can we maybe switch the detrimental inflammatory microglia to a pro-neurogenic M2 type microglia by doing stem cell transplantation? So we started investigating the neurogenic zones, subventricular zone and the hippocampus. And going fast forward, we looked at what soluble factors are secreted by neural stem cells or are secreted by microglia in this environment of stroke. What we found is that actually the neural progenitor cells in the neurogenic zone secrete a lot of VEGF upon hypoxia or as a response to hypoxia. And VEGF actually can really change the function of microglia, both in terms of cell proliferation, but also in terms of phagocytic activity, in terms of clearing debris and helping restoration or regeneration by secreting pro-neurogenic factors. The other way around, can microglia secrete factors that help neural stem cells in the brain? And this was a PhD student in my lab now in Switzerland. The work before was from Stanford. And he found that microglia actually secretes growth factors that will help stem cells proliferate, differentiate, and survive in the dish. So he did these co-culture experiments. And if he eliminates the microglia with this anti-CD11b antibody, so he withdraws the microglia, the effect on these neural stem cells in the dish is actually lost. So there is actually a bilateral communication between stem cells and microglia in the brain. Very interesting concept, and we're carrying on that line of research. So now some current projects. So we're 3D printing microglia with a bio 3D printer to actually have a very structured way of looking at oligodendrocytes and look at the effect between neural stem cells, microglia, and myelinating oligodendrocytes. So trying to mimic this interaction between the different brain cells in response to transplantation of neural stem cells. All of this just to understand better the mechanisms of action of repair. We did a review analysis in the literature on cell transplantation for neonatal stroke, and we see actually that all of those studies have immune modulation, so modulation of microglia and other immune cells as one of the very important effects after cell transplantation. So in conclusion to the intravascular cell delivery, we can say that cells home to the lesion, they enhance endogenous neurogenesis. They have an effect on white matter repair, protection or regeneration is still a bit unclear, and they have a very strong effect on immune modulation. All three mechanisms of action important probably to have a functional benefit after transplantation. So a current project is hypothermia. How does hypothermia relate to all of this? Hypothermia, we know it as a neuroprotective agent. We were actually wondering if there's a molecular mechanism that would connect hypothermia and endogenous repair. So cooling the brain is well known for all of you. And there is actually a molecule, which is a cold inducible RNA binding protein that can be enhanced upon cooling of the brain. And again, I will not go into all the details, but it was shown very importantly that it can promote structural plasticity, reduce apoptosis, and stabilize mRNA in the communication in the injured brain. So in this study, what we did, we tried to demonstrate that there is actually expression of this cold inducible protein in the neurogenic areas. And we studied the subventricular zone and the subgranular zone of the hippocampus, both areas that actually create neural stem cells. And if you upregulate through hypothermia or injury this cold inducible protein, you actually have a reduction of the lesion size, okay, lesion size after stroke. But if you have a knockout for RBM3, this effect is lost. So going fast forward, what we saw in this study is that upon hypoxia, if you have an upregulation or a downregulation of RBM3, you have actually an enhancement of endogenous neurogenesis as one of the neuroprotective and neuroregenerative capacities. And the molecular pathway that we found out is that actually RBM3 conjoins or forms a bind to IMP2 and leads to an upregulation of insulin growth factor 2, which is a very strong neurogenic factor that can lead to proliferation of neural stem cells, and at the same time RBM3 can inhibit apoptosis. So we have two effects. One is neuroprotective, one is actually neuroregenerative, and we're testing now different drugs that would selectively upregulate RBM3 in the brain as a neuroprotective and neuroregenerative approach. Because hypothermia, as we all know, has its own problems in terms of negative side effects. So we're investigating now RBM3 in microglia and so forth. So this is part of current studies. Before I go to the clinical trials, here's our friend Daniel, who was actually defending what we call in Europe the habilitation. It's when you become an associate professor, a very nice event where the dean of the medical school was talking highly to Daniel, and shortly after actually defending his associate professorship at the University of Basel, he unfortunately left to the US. But this was a very nice last event of Daniel in Basel. So moving forward to the clinical trials, these are the current concepts for clinical trials in patients. So we have different routes of administration. We have intraperitoneal, not so good for patients, but very good in rodents. But we have intravenous and intraarterial cell injection. We have, of course, intracranial transplantation. And a new mode of transplantation is intranasal, maybe more for kids than for adults, but a very interesting mode of transplantation. These days, we use neural stem cells, adipose-derived stem cells, umbilical cord-derived stem cells, and bone marrow-derived stem cells in different clinical trials. And you will see that there is a lot of trials going on, both with autologous stem cell transplantation, both with allogeneic transplantation. So if you go to clinical.gov, clinicaltrial.gov, you will see there's a lot of studies going on. And I will just mention a few that I think are kind of the key or landmark papers in neural stem cell transplantation for stroke. The first one published in 2005, intraparenchymal transplantation. This was Doug Konciolka and Gary Steinberg, so Pittsburgh and Stanford doing that trial together. And they actually did show in a chronic stroke model, they transplanted 5 to 10 million cells, that they actually had a functional recovery in hand motion. But the trial was, of course, a phase 1/2 trial, was not powered to demonstrate a statistically significant difference between the two groups. But it was a first trial actually demonstrating a functional recovery in patients. The second important trial was this PCIS trial, published in 2016, now in Lancet, up to 20 million cells, again, chronically, so in chronic situation after stroke, and they had safety and neurological improvement in terms of a reduction of the NIHSS score after 2 years in 13 patients. Then we have two important trials with intravascular stem cell delivery, both intravenous or intraarterial. This was the MASTERS trial and the IBIS trial, this year out of Spain, this year a big consortium out of the UK and the US, where they transplanted up to 1.2 billion cells in 33 centers, 129 patients. It was safe, but not efficacious. And here a trial of intraarterial bone marrow-derived cells, up to 5 million cells per kg, acute, in 76 patients, safe, but not efficacious. So there's still a lot of frustration going on in that field. Here another study by Gary Steinberg from Stanford, who has really pioneered the field, transplantation chronically, and what he showed here, up to 12 months after transplantation, a gradual decrease here in the NIHSS score. Now, is this natural history or is it transplantation? It's not a randomized trial, so this cannot be answered. But just based on historic data, these patients were not supposed to have such a recovery in a chronic phase, so past 3 months after stroke. Here, Sean Savitz out of UT Texas, safety in the RECOVER stroke trial with bone marrow-derived stem cells, but no efficacy in this large transplantation trial. And finally, a very recent study out of Japan, published just now in JAMA Neurology, where they transplanted in a randomized trial 206 patients, whereas 140 received these mononuclear bone marrow-derived stem cells in the acute phase after stroke, intravenous injection, and they looked at the NIHSS score. Patients were included if they were between 8 and 20 score. And what they found was a trend towards improvement in the stem cell transplanted patients versus the placebo at 90 and 365 days. So an improvement in MRS 1 to 4, but again, no statistically significant results. But there was a trend at least. So raising the hope that if you slightly adapt the trial and the cells, potentially there is a role for such a treatment. But there is no definite study that would prove beneficial effect. And Gary Steinberg just last week at the AANS presented again a smaller study on 18 patients. And what he found a recovery 6 to 12 months after transplantation in patients that were 2 years, 6 months to 5 years after stroke, in an average 2 years after stroke. So patients in which you did not expect to have any improvement actually did show an improvement at 6 to 12 months after cell transplantation. So it's a weak signal, but again, there is a signal. So if we take this together in this meta-analysis published in 2019, so not including the newest studies, we see that actually, if you look here at the modified ranking score or at the NIHSS, all the studies actually favor cell transplantation versus best medical treatment for these two factors. And also, if you look at the different routes of delivery, intravenous, intracerebral, intrathecal, and intraarterial, they favor cell transplantation over conventional treatment. The effect is weak, and we see that the effect has been stronger in intracerebral transplantation than in intravascular, but intravascular, of course, is more feasible and more scalable. So to conclude, what are the persisting challenges? One is cell dosing. We still don't know exactly what is the right dose. You saw the dose goes from 5 million to 1.2 billion, depending on transplantation mode, but there's no definite answer on dosing. There is no definite answer on timing. Should you inject acute or chronic? Maybe you should do both. What cell type? Because we have the mesenchymal stem cells, we have the neural stem cells. Which one are the most potent? Still open for discussion. And as I mentioned, the route of delivery, what is the best route of delivery? Maybe a combination, acute intravascular, but chronic, which is more geared towards restoration, intraparenchymal. These are all open questions. Another open question is logistics, how to transport. And here just a final anecdote. In 2011-12, I ran a transplantation trial for spinal cord injury. Now you imagine those cells, they are produced somewhere in a lab. In this situation here, for the stem cell transplantation to the spinal cord, the cells were prepared in Palo Alto in a company called Stem Cells Inc. The FDA did not accept the trial. I suggested, why not do the trial in Switzerland, where the Swiss EMA, or what's called the Swiss EMA, actually accepted to do the trial. Now you had to transport those cells from California to Switzerland and transplant them within 24 hours. You can imagine the logistic nightmare. So by chance, because Roche bought Genentech just at that time, there was a new Swiss flight being installed. So every day there was a Swiss flight from Zurich to San Francisco direct, not some indirect flight. So we were able to actually manufacture the cells, pack them in special container with temperature sensor and movement sensor, ship them to Zurich. They go to customs. They arrive at 10 in the morning. They go to customs, and at 2 in the afternoon, I was able to transplant them in patients. And like this, I transplanted 12 patients with spinal cord injury. But just to say, it's not a drug that you can dry pack and ship in the whole world. The logistics around it is actually quite complicated. And that's why also the financial burden is a problem. Big pharma doesn't want to invest so much into stem cell. It's not a drug that they can scale up and sell. Produce cheap and sell expensive. It's a biologics, and biologics are inherently more complex to actually produce and scale up. So there is still hurdles. So it was just the European Stroke Awareness Day with the ESOC meeting in Basel, and we had a meeting actually on how should we carry on. And I will end up by saying that there is a lot of thoughts how it could go on. you know, what are the best cell types, timing, and so forth? How could we engineer those cells to be more efficacious? And how could we deliver the cells quickly after stroke, have image guidance, but also imaging to actually track the cells? So there's a lot of thoughts going in. And we have a consortium between Europe and the US, sponsored by the NIH, to actually define steps. And that's why it was called the STEPS meetings. The last one is a while ago in 2019, where we defined what are the next steps that should be taken in terms of the research on stem cell transplantation. And with this, I would like to end. I hope I was able to bring you some concepts between cell transplantation, cell repair, trophic support, and restoration in the brain. And of course, we all hope that this topic will go on and hopefully be fruitful in the next decade. Thanks a lot.
Wow, amazing talk. A lot of it perfectly illustrates the decades-long journey with lots of efforts, the complexity of the goals and tasks, and also the challenges, but also offers hopes in the future for folks, for people who suffer the stroke, cannot be rapidly reversed acutely with residual deficits, residual injuries. What's lying ahead with the hope? Thank you. Thank you for an amazing talk. Any questions in the audience? Dr. Zumafan, of course.
Of course, amazing talk. Thank you so much. I think we all agree on the huge burden of stroke and the importance of a recovery. All the companies are currently investing in that field. Here in Brooklyn, down the road, there is a company called Synchron producing brain implants, brain to computer interfaces, Neuralink, Elon Musk heavily investing. I think it's the field that's taking off.
Yeah.
And you louder think about your kids 50 years down the road. Will they have stem cells, or will they have a robot, or will they have both?
Good question. If I had this crystal ball, I would answer you correctly. But I think the one important thing you mentioned is that neuromodulation, of course, is one of the big topics. And I think we will hear talk about neuromodulation also. And one of the concepts that is being studied is combination of neuromodulation. So I'm not talking about human-machine interface. I'm actually talking about enhancing plasticity by neuromodulation, by electrical stimulation of the brain. So the combination of stem cells and neuromodulation could be one way to go, and it's being investigated preclinically. So I think it's an exciting field. So maybe they will have both.
Okay, so I'll wait for everybody else.
Dr. Guzman, that's quite an impressive talk. The best stem cell lecture I've ever heard. It's very enlightening, very, very helpful.
Thank you.
Before I ask you the question, I'm going to answer your question on that 50 years from now, because at Barrow, we're the first one installed the Neuralink. Now that patient was a quadriplegic, but the brain is okay. So the Neuralink was able to capture his thoughts and then start to manipulate the computers. So my thinking is that for that, you need a normal brain to have a link. If you have a really infarcted brain, I'm not sure there's these hair thin wires can go anywhere, but maybe they'll change that. But that's what I observed from that at the Barrel. But my question to you is that we're also doing a little bit hypothermic work. I've seen that the RNA binding protein is being promoted under hypothermia. So in your laboratory, my question is, how cold do you induce that hypothermia? To what degree, and how long do you keep that coldness?
Yeah.
Thank you.
Yeah, thank you. Yeah, I think hypothermia is a huge topic, and I know that there is work going on at Barrows, and there was a lot of work also going at Stanford while I was there. So it's mild hypothermia, very important. The deep hypothermia at 30 degrees is detrimental and actually has a lot of negative side effects. So it's 33 to 34 degrees. And experimentally, because we're working now mainly on cells, is actually for 24 hours. And the strongest effect is after 48 hours. So even when we stop hypothermia at 24, we have an upregulation of the protein that peaks at 48 hours. And in the animals, usually it's 1 to 2 hours hypothermia. That's it. Mild hypothermia. Yeah.
Gary Friedman from Brookdale Hospital. The stem cell transplantation for Parkinson's disease, did you do them in the caudate nucleus or did you do them in the mesencephalon?
In the caudate, but it's a good question because it seemed too risky to go to the mesencephalon. So we transplanted where the dopamine actually is being used. But yes, it was in the striatum. Yeah.
Thank you.
Amazing talk. The the other thing that we forget is that the same exact principles could be used for trauma.
Yeah.
And that's even a bigger burden because it tends to impact much younger people and could potentially be even more successful, especially like pediatric trauma and trauma in the 20s, because there are many patients that are debilitated. But to his question, I think that it's probably going to be a combination of both. I think that it's going to depend on the disease. Spinal cord injury may be completely different than trauma and stroke. And then solving the piece of how to get it there is probably, or what is going to be the most effective way to get it there.
Yeah.
I mean, because if you think about it, optimally, the best will probably be, imagine if you could do a thrombectomy and deliver the drug right at that same time.
Yeah.
Right before you even have all the cell loss. And then that potentially opens up the possibility of not stem cell transplantation, but, is there a way to regenerate what is not completely dead yet? And
Yeah, I think one of the visions, of course, is, you do your thrombectomy, you open blood flow, you can inject, do allogeneic transplant. You cannot do autologous at that time, but allogeneic. And then later on, maybe in a second time, do autologous cell transplantation.
multi-stage too.
Yeah, multi-staged, I think. And trauma, of course, trauma is understudied in general, I would say, and even more understudied in research because it's a dirty model. Stroke is already a dirty model. Trauma is even a dirtier model in terms of molecular mechanisms happening. So it's understudied. Yeah.
Great. What always puzzles me conceptually for these cell delivery strategies is that we are delivering these cells that are potentially pluripotent and can differentiate into any cells that are needed or also integrate into this kind of damaged area. That's the hope. How do we know they have the right cues to differentiate into the right cell types and in the right place? Just like you parachute the paratroopers behind enemy lines in disaster areas, there are no supporting system, no GPS. How do you know where to go?
Yeah.
Conceptually, how do we overcome that barrier?
Yeah, so this is an area of intense research. But one point is that with the hypothesis of trophic support, it seems actually not to matter so much. And when you do the biodistribution studies, you actually see that 99% of the cells disappear within 7 days. So they do actually not integrate and not differentiate. They're more viewed like they respond to cues, molecular cues, like inflammatory cytokines and so forth, by releasing factors, but they do actually not integrate and differentiate into the cells, into brain cells. So the concept of the trophic support has really freed us from the idea that the cell needs to develop into a brain cell. This is different for the transplantation at the chronic stage of neural stem cells. And there, in fact, the cells does get the cue in terms of what do they differentiate into? Neurons, astrocytes, oligodendrocytes, interestingly. The exact molecular cues are not really known though. But they have a 3 lineage differentiation. And what was found is that depending on when you transplant them after stroke, they will preferentially go into astrocytes if you transplant them early, and they will preferentially go more into a neuronal and oligodendroglial differentiation if you transplant them late. In the sense that almost the brain is looking for regenerative capacity and differentiates the cells in the cells that are needed potentially. But the exact molecular cue is still open.
That's very interesting. How do we know those clinical recoveries, which is the foundation for the trophic theory, is actually derived from these stem cells that were delivered and not from intrinsic residues, just like you were saying from the Dr. Steinberg's trial, that recovery is clinically we did something, but how do we know it's natural results?
So of course, experimentally, we can have good controls, right? So they get, saline, or they get IgG if we want to just test for one protein. So they get IgG versus cells. So we do see that there is a significant difference in animals that get cells versus getting some control. But
in humans.
Well, exactly, in rodents. Now in humans, you're completely right. It's different. How would we know? And one of the big problems is we have great biomarkers in rodents, right? We have no biomarkers in humans. We have no biomarkers that would prove that the cell did the difference. Now, we do have slight difference in the randomized trials between saline and cells, but it's not statistically significant. But there seems to be a difference. And the only difference these patients had, they were pretty well balanced in terms of, stroke size, location, and so forth. The only difference is getting cells or not. But there is no biomarker to actually prove what has the cell done. And people have done, plasma and everything. They look for cytokine expression in the blood. So there's a lot of research just into biomarkers. There's no definite answer.
Thank you.
Yeah.
Fascinating.
Thanks a lot.